Hi, this is Larry Markham, Professor of Pediatrics at Indiana University School of Medicine and Riley Hospital for Children. I'm going to talk today about our FDA-sponsored study of Ifetroban for ambulatory and non-ambulatory patients with Duchenne muscular dystrophy. I have no personal disclosures or conflicts of interest other than being the principal investigator for this trial. Ifetroban selectively blocks the thromboxane prostanoid receptor, and as such, this receptor is expressed on many cell types, including platelets, immune cells, smooth muscle, and cardiomyocytes. This activation is pro-fibrotic in the heart and lungs, and as Ifetroban prevents this fibrosis and inflammation. Ifetroban is an oral medication which is administered once daily, and there are several ongoing studies using this dosing. Safety is well established with over 1,300 clinical trial participants in over 30 clinical trials. This cartoon depicts where Ifetroban's mechanism of action works, and as an example, you can see at the top, ASA or aspirin, that is where aspirin works, and Ifetroban blocks all of the receptors which activate pro-fibrotic pathways. This inflammatory response and fibrotic pathway is activated in Duchenne muscular dystrophy. When you have a DMD mutation, you have an abnormal or absent dystrophin protein, which impacts the muscle cells themselves and leads to a cascade of events leading to inflammation, fibrosis or scar tissue, and then thinning of that heart muscle. That results in changes in ventricular volume, increased wall stress, and abnormal relaxation of the heart, which leads to heart failure. So the thinking behind this is that if we can impact this inflammatory and scar tissue formation, maybe we can impact the heart failure associated with Duchenne muscular dystrophy. I'd like to thank Dr. Carrier and Dr. West at Vanderbilt, who we collaborated with to acquire this data, which was published last year. So they took different groups of muscular dystrophy mice, and this data shows survival in panels A and C, and then measures of heart muscle function in B and D. The first group was the double knockout mice, and those treated with Ifetroban had better survival than those who did not. The same is true for the MDX telomere mice, where you can see the blue line showing the treated mice had markedly improved survival compared to the untreated mice. Panels B and D show measures of fractional shortening, cardiac index, and stroke volume, where the blue panels are the treated mice. You can see that cardiac index and stroke volume improved after treatment with Ifetroban. They also studied cardiac dysfunction and survival in the limb-girdle muscular dystrophy model and had similar results as to the MDX mice, where there were improvements in fractional shortening, ejection fraction, as well as survival. Ifetroban also normalized ANP and LV systolic pressure in limb-girdle muscular dystrophy mice, and the treated mice are shown in the blue panels. Importantly, Ifetroban blocked the pro-fibrotic pathways in the heart muscle in particular. And so in these panels, you see sections of heart muscle tissue, and in the top panel, in the second and third heart, you see vehicle-treated mice who have the purple scar tissue, which is shown by red underneath. This was markedly improved in the Ifetroban-treated mice. Measures of fibrosis by counting collagen-positive areas or phosphorylated SMAD show significant improvements with treatment of Ifetroban. Again, this data has been published by Dr. Carrier and Dr. West last year. So this led us in our work in collaboration with the Marlin family and their foundation, Fight DMD, to develop Ifetroban as a therapy for Duchenne muscular dystrophy cardiac dysfunction. So the Fight DMD trial is the first DMD trial awarded an FDA orphan product grant. The inclusion criteria for the trial include males 7 years and older with diagnosis of Duchenne on stable corticosteroids, or those who have elected not to use corticosteroids. We would like individuals to have stable cardiac function over the last 12 months, and that is defined as a change in LVEF less than 15% and no heart failure admission to the hospital. Those individuals should have an LVEF greater than 35% by MRI or echocardiogram, and having heart muscle scar or fibrosis is allowed. Individuals can continue their medication such as ACE inhibitor, beta blocker, angiotensin receptor blocker if they were started 3 months or more from the first trial dose of medication. Spironolactone or eplerenone are allowed if started greater than 12 months from entering the trial. Some specific exclusion criteria include severe ventricular dysfunction with an EF of less than 35%. Participants who are in another therapeutic clinical trial, we will need to discuss the timing and if it's possible to enter this trial. Because of its impact on platelets and bleeding, anyone with a known bleeding disorder or receiving anticoagulant therapy will be excluded. Anyone with allergy to gadolinium or known renal dysfunction will be excluded or unable to undergo a cardiac MRI. And so this diagram outlines the study design of enrollment for 48 boys to evaluate the efficacy, safety, and pharmacokinetics of Ifetroban. It will be daily oral dose for 12 months. 2/3 of subjects will be randomized to treatment. 1/3 will be placebo. There will be 3 in-person visits over the 12-month time period to include MRI, pulmonary function tests, quality of life surveys, and measures of skeletal muscle strength. Then there will be an open label extension where placebo participants will have the opportunity to receive Ifetroban. Key assessments will be, as we talked about, the pharmacokinetics. We have explored a novel technology where we will use a finger stick similar to measuring blood glucose. The dosing measurements for pharmacokinetics at 8 and 24 hours can be performed at home, as well as the day 7 pharmacokinetics. So some key eligibility and study features: all boys greater than 7, regardless of mutation type, ambulation status, or steroid use, are eligible. There is no upper age limit, and some standard of care medications will be allowed. There is no biopsy or 6-minute walk test required. At present, there are 5 sites which are beginning to be up and running. My hospital in Indianapolis, Vanderbilt in Tennessee, Children's National in DC, Emory in Georgia, and then UCLA in California. As you can see below, there is a website, fightdmdtrial.com, and individuals can reach the study group at FightDMD at cumberlandpharma.com. With that, that concludes my presentation, and I would like to thank Cumberland Pharmaceuticals, the FDA, FightDMD, as well as Parent Project for this opportunity. Thank you very much.